Today we'll talk about the most important system of our body. You know that the nervous system is the most powerful and important system in our body because among the 11 systems that we have, 2 systems are called the control systems. You know that the nervous system and the endocrine system. And you also know that the nervous system is more powerful than the endocrine system. So we'll talk about the nervous system. First, in the first half of the lecture, we'll talk about the following things: functions of the nervous system. Then we'll see the divisions of the nervous system, how we divide the nervous system into different groups. Then we'll talk about the cells found in the nervous tissue or in the nervous system, different types of cells. In the nervous tissue, only 2 different types of cells are found. The main cells are called the neurons, and the supporting cells are called the neuroglial cells or neuroglia. So first we'll talk about the supporting cells, that means the neuroglia. There are different types of neuroglial cells. So we'll see how they look like, and we'll briefly talk about the functions of different types of neuroglial cells. Okay? Then we'll talk about the main type of cells in the nervous tissue, that's the neuron. So we'll talk about the neuron, we'll talk about the parts of a neuron, different types of neurons and functions of neurons. Then we'll see what does mean a tract, what does mean a nucleus, what does mean gray matter and white matter. We'll just see what are those structures are present in the nervous system. Those structures, tracts, nuclei, gray matter, white matter, are seen very often in the nervous tissue or nervous system. So we'll see what are those structures. We'll see in the nervous system. Then we'll talk about the reflexes and the parts of a reflex arc. What is reflex? Very simple. If I put flashlight on your eye, your pupil constriction of pupil occurs. That's a reflex. If I hit with a small hammer on your patellar ligament here, you know that the leg will shoot forward, kick forward. That's a reflex. Okay. So what are the parts of the nervous system involved in that reflex? That's the reflex arc. So we will see what does reflex mean and the components of or parts of a reflex arc. So in the first half of our lecture today, we'll talk about these things. First, the functions of the nervous system. Our nervous system performs thousands of important functions. Just example, you see with your brain. Actually, the eye just only captures the picture, but you see with your brain. You smell with your brain because the smell signal goes to the brain. You hear with your brain. You perform the task. You move your body parts. That is done by the brain cell signal. Right? So we can give thousands of examples that those are the functions of your nervous system. Now, we can classify those functions into 3 groups or 3 types of functions. What are those? Number 1, sensory input. What is sensory input? For example, you are thirsty, you are looking for water. So when you see a glass of water, that picture will be taken by your eye and will be sent to the brain. So that's the sensory input that's getting in from outside. Okay. Then after that, what is next? Your brain will interpret. What your brain does? Just think simply. Your brain, first thing your brain will do is it water or something else, right? Then your brain will also think if it is water, is it clean water or not? So many things your brain is quickly doing, right? How far the glass is, your brain has to measure it, right? And then your brain will interpret those, read out those signals. That part is called integration. So sensory input is fast, the signal, the picture will go to your brain, that's the sensory input. Then your brain will interpret and try to measure all those information, and that's the integration. And then your brain will send signal to your muscles to move your hand to grab the glass. Make sense? That's the motor output. Motor is the movement. Okay? Make sense? So we can divide, you see, these functions of your nervous system into 3 groups. Sensory input, integration, and then motor output. Another example. If something very hot touches your skin has temperature receptors. So your skin will send that signal to the brain. And your brain will do what? In this instance, your brain will decide if the hot is too hot or it's okay, right? If it is too hot, what you have to do, right? All those things will be interpreted by the brain. The sensory signal should be interpreted by the brain. Then your brain, if the brain decides that, thinks that it is dangerous, too hot, can burn your skin, your brain will send signal to your muscle to move your hand away. But all those things will be done very fast, right? Something very hot touches your skin, you will move your hand away. Very fast, right? So you see, the sensory signal is temperature signal is going to your brain. Your brain is interpreting that. That is also the function of the nervous system. And then brain is sending signal to the muscles to move your hand away. That's also the function of the nervous system. That part is called the motor output, getting out from the brain. So we can divide all the functions of the nervous system into these 3 groups. So the example I have given you, I have been shown here. Sensory input, then brain interprets, that's the integration, and motor output is going to the muscle. Divisions of the nervous system. We can divide the nervous system into 2 groups. Central nervous system and the peripheral nervous system. Those are the 2 parts of the nervous system. Central nervous system includes the brain and the spinal cord. So if I ask you in the test that which organs or structures are the parts of the central nervous system, the brain and the spinal cord. And peripheral nervous system includes spinal and cranial nerves. So outside of the central nervous system, you have a brain and spinal cord. These 2 are the parts of the central nervous system. And then from your brain, a number of cranial nerves arise. And you have seen, when you have seen the base of the skull, you have seen many holes, right? You remember many foramina at the base of the skull, floor of the skull? Some of those for the blood vessels, but many of those for the nerves. So from the brain, those nerves arise and goes down, get out from the brain. Those are the cranial nerves. Cranial nerves. Okay? And now, from both sides of your spinal cord, those nerves arise. Those nerves are called the spinal nerves. Okay. So cranial nerves arise from the brain, and spinal nerves arise from both sides of the spinal cord, and those are the parts of the peripheral nervous system, cranial nerves and the spinal nerves. We have 12 pairs of cranial nerves. Okay, 12 pairs of cranial nerves, and we have 31 pairs of spinal nerve. Okay, so those are the peripheral parts of peripheral nervous system. Okay. Now, peripheral nervous system has two functional divisions, afferent and efferent. One starts with A and another starts with E. What's the difference? Afferent nerves take the signal towards the central nervous system, and efferent takes the signal out from the central nervous system. So let's see here. This is one cranial nerve, and if that cranial nerve is taking signal from outside to the brain, that's the afferent. It starts with A. And if this cranial nerve is taking the signal out from the central nervous system, that's the efferent. Okay? So afferent will take the signal towards the central nervous system, and efferent takes the signal out from the central nervous system. Okay? Now, let me give you an example and you will understand. So something very hot has touched to your skin. From the skin, the signal is going to the spinal cord, and from there, it is going to the brain. So is it afferent or efferent? Afferent. It starts with A, because going towards the central nervous system. Make sense? Then your brain will do what? Interpret, right? And then brain will send signal to the what? Muscles. So that signal is getting out, right? From the brain and the spinal cord. So that is what? Afferent. Starts with it. So you got the idea that afferent is going towards your central nervous system, efferent is getting out. Another way you can remember, afferent starts with E, that means arriving, and efferent exiting, getting out. Okay? So A and E. Okay. Now, the afferent is sensory. The signal, all the signals, those are going towards your central nervous system. Those are sensory signals. What does it mean? What are the sensory signals? Sensory signals are touch, pain, temperature, visual signal, smell, hearing or audition. All those are going towards your central nervous system from outside. Those are sensory. Okay? Those are going towards the central nervous system. Those are sensory. And those are getting out. For example, the signal is getting out to your muscle. You are moving your hand away. That is motor, because movement is the motor. Remember, contraction or movement is the motor. So sensory is afferent, motor is efferent. Okay. Same example. When something hot touches your skin, that's a sensory, because I said that pain, touch, temperature, all those are sensory signals. So that sensory signal is going towards your brain. That means sensory is afferent, and then brain is sending signal to the muscle. You are moving. That's the motor. Motor is the movement. Okay. So motor is efferent. Now, afferent could be somatic afferent and visceral. That means sensory could be somatic sensory and visceral sensory. What's the difference? So pain, touch, temperature, those are what? Sensory or motor? Everybody? Sensory. Very good. I just need to make sure that you are understanding, then I can go to the next. Okay, so all those are sensory. So you see, if you feel pain in your skin, that pain signal is going towards your central nervous system. That's sensory, right? That's sensory because pain is sensory. You also feel pain inside of your body, like stomach pain. Makes sense? Heart pain. You feel that, right? So the pain that you feel in the outside part of the body, that signal is going to the central nervous system. Also, pain signal is going from the internal body organs to your central nervous system. The somatic means when the pain sensory signal is going from outside structures of the body, for example, from your skin, muscle, those are somatic. Somatic indicates the outer body structures. And viscera are your internal body organs, for example, your liver, your kidneys, your lungs, right? Your heart, those are viscera. So you see that sensory signal can go to the central nervous system from both from the outer structures of the body as well as from the internal viscera. So that's the difference, okay? Somatic is from the skin, muscle, joint, and visceral from the internal body organs, for example, your heart, lung, kidneys, liver. Okay, so that's the difference. Okay, now motor could be two different types. Efferent or motor could be also two different types. What are two different types? Voluntary and involuntary. Example, same example. When you move, if I ask you, move your hand, you can control that movement, right? You can move your hand out. Now, if suddenly something very hot touches your skin, you don't need to think, you will automatically move your hand away. Make sense? You will automatically your hand will move out. So when you decide and send signal to your hand that I will move my hand willingly, that is the voluntary. And when it happens automatically, that's the autonomic. So voluntary and autonomic. And voluntary is the somatic and involuntary is autonomic. Okay? So for example, if I ask you to move your hand away, you can control your muscle, right? You can do that. Now if I ask you to stop your heart is a muscle too. Can you do that? Stop your heart, unless you kill yourself. You cannot do that, right? So you see, both are muscles. Your skeletal muscle, you can control willingly. But your cardiac muscle, you cannot control, right? You have no control on your cardiac muscle. You cannot stop it. So one is the somatic is voluntary, that means the skeletal muscles, and autonomic are involuntary. Those you cannot control. So those are two different types of motor. Okay. Now the autonomic has two divisions, two functional divisions: autonomic, sympathetic, and parasympathetic. Sympathetic, just know that when you activate your sympathetic division of autonomic nervous system, what kind of changes occur in your body? The simplest way is when you get excited, what kind of changes you see in your body? Those are the results of activation of your sympathetic division of autonomic nervous system. So when sympathetic nerves are activated, you feel excitation. So example, what will happen in your heart if you stimulate the sympathetic? Heart rate will increase, go up. Yeah. So force of contraction will increase. Sweating will occur. Muscle tone will increase, right? So all those things happen when you get excited. So those are the results of the activation of sympathetic division of the autonomic nervous system. Now, parasympathetic is just opposite. when you are in very comfortable, relaxed condition, like you are sitting on a couch and you are watching golf, beautiful golf course, okay, like green and nice trees, lake. So unless you are a golfer, then the lake or bunker is not good. The sand is not good, then you will get nervous. So yeah. So otherwise, if you are not a golfer, you are watching golf, beautiful, everything looks good, right? So in that case, what will happen? Your heart rate will slightly decrease, your blood pressure will slightly decrease, your muscle tone will decrease, okay? Your muscle will be relaxed. So those are the results of activation of parasympathetic division of the autonomic nervous system. Okay? So just know, if I ask you that if you activate the sympathetic nerves, what kind of changes will occur? And if you activate your parasympathetic, what kind of changes will occur? That's the difference. Okay? In one sympathetic, in sympathetic you are excited, in parasympathetic you are relaxed. Okay, now we'll summarize the talk that I have just given so far. So we'll see here, this is a nice picture. You can see the division of the nervous system. So here you see the brain and the spinal cord. Those two structures belong to the central nervous system. So, central nervous system includes the brain and the spinal cord. And then, peripheral nervous system includes the spinal and cranial nerves. Those are outside of the central nervous system. Then, the peripheral nervous system is divided into somatic or afferent and motor or efferent. Somatic is sensory. So, going towards the central nervous system. So if you see the sensory or afferent, if you see the sensory, the blue lines going signal towards the central nervous system. Okay, this way. Now, somatic, sensory and visceral. Sensory could be somatic sensory and visceral sensory. Somatic, I have given you the example from the outer structures of the body. Those are somatic. Just remember, somatic indicates the outer structures of the body. For example, your skin, your muscles, your joints. If you feel pain in those structures, those are what kind of sensory? Somatic sensory. Now, the visceral sensory is, visceral means internal organs. Just remember for this purpose, that visceral indicates the internal organs. For example, your stomach pain, your liver pain, kidney pain, lung pain, heart pain. Those are visceral sensory. Okay? So somatic sensory and visceral sensory. Those are the different types of sensory. One is getting in from the outer structure, another is getting in from the inner body structure. Then motor is divided into the somatic and autonomic. Okay? Then autonomic is divided into two types, sympathetic and parasympathetic. And I have talked about sympathetic and parasympathetic. What will happen if you activate those two divisions of the autonomic nervous system. And you see the red lines are showing that signal is getting out from the central nervous system. Okay? Getting away from the central nervous system. So that's the motor. So blue lines are sensory, red lines are motor. Okay, now we'll see the cells, different types of cells present in the neural tissue. As I have already mentioned, that we see two different types of cells in the nervous tissue. The main type is called the neuron. So neurons are the main cells of the nervous system. And supporting cells are called neuroglia or neuroglial cells. if I ask you in one sentence, tell me the function of a neuron. The function of a neuron is production and transmission of electrical signal. So electrical signal is produced or generated and transmitted by a neuron. Okay, that's a nice simple sentence. It's electricity, produce electricity and transmits electricity. Neuroglial cells. There are different types of neuroglial cells or supporting cells we have, and they perform different functions. So in next few slides, what I will do, I will go over the important neuroglial cells, and I will do two things. You need to remember, there could be a few questions from here. How they look like, and number two, just one or two important functions of those neuroglial cells. Okay, you have to remember. So what are the functions the neuroglial cells perform? Neuroglial cells migrate towards the injured neurons. So, if any injury occurs in your nervous system, the neuroglial cells can move towards the injury, site of injury. Why they need to move there? Because you know that when injury occurs, many cells die. So those dead cells should be engulfed and removed from there to clean that site, number one. Also, neuroglial cells will move there if there is any microorganisms. Sometimes microorganisms cause infection, you know that. So in that area, what will happen? The neuroglial cells, some neuroglial cells will move there and will engulf the microorganisms. Okay? So that is one important function. Another is, so I have mentioned already these two, guide migration of young neurons. What is that? That happens more in early life, when your brain is developing. What happens? You know that in a fetus, in an embryo, the brain tissue is a small amount of brain tissue. Then what happens? From there, different structures, brain structures are formed. Okay? So you know that in very early life, we have only few cells, right, in an embryo. Then from there, the cells move to different directions and form different parts. So the young cells, these cells, brain cells, move towards different directions and form different brain structures. For example, your cerebrum, your cerebellum, and thalamus, hypothalamus. So just know that those movements are guided by some neuroglial cells. Neuroglial cells will tell the neuron that you move this way, you move that way, you move in another direction. So that is another. Control the chemical environment. Some neuroglial cells can secrete some chemicals and maintain the chemical environment. And insulation is another important function. Insulation is, you know, that electrical wires we usually insulate, cover. So that insulation does two important things. One is it lets the electrical signal travel faster. So the electrical signal will move fast and will prevent the loss of signal. So since you are covering the wire, the electrical signal will move fast, number one. Number two, the loss of signal will be reduced. So insulation is done by some neuroglial cells, because you know that the neurons transmit electrical signal. Transmission of electrical signal is a function of the neuron, right? So the electrical signal will not get lost, number one, will travel very fast if insulation is done. And neuroglial cells do that, okay? So I explained these things so you will remember better. But just remember the name of the functions. You don't need to remember everything I said. I just mentioned those things just to make it understandable to you. Now you will be able to tell. If I ask you, tell me 3 or 4 or 5 important functions of neuroglial cells, you will remember those better, right? So those are, in general, the functions of the neuroglial cells. Now, in next few slides, what I'll do, I'll go over individual neuroglial cells and tell their 1 or 2 important functions. So, what are the important neuroglial cells we find in our nervous system? Astrocytes, microglia, ependymal cells, oligodendrocytes, satellite cells, and swan cells. So those are important neuroglial cells. Now, here you see the picture of an astrocyte. Astrocyte, site is cell, and astro indicates what? Star. Astro, you know astronomy. We use that term, astronomy. So astrocytes are star-shaped neuroglial cells. And astrocytes are the most abundant neuroglial cells, most commonly found neuroglial cells. So, 2 information you got from here, I may ask you. One is, if I ask you which neuroglial cells look like star, that's the astrocytes, and which one is the most abundant type of neuroglial cells, that's also the astrocyte. Okay? Microglia. Microglial cells have many long processes arise from the body. So if you see a number of long processes originated from the body, and microglial cells main function is phagocytosis, and by phagocytosis it provides defensive function in the nervous system. That means they can engulf the microorganisms, and I have mentioned that engulfing microorganism is one important function of the neuroglial cells, right? So this type of neuroglial cells will do that. Will engulf the dead cells as well as the microorganisms, and will provide the defensive function, okay, by killing the microorganisms. And that's why it has many long processes, long hands. They can engulf easily the microorganisms. Ependymal cells. Inside your brain, you have a number of cavities. For example, if this is a brain, you have, inside the brain, you have cavities, and these are called ventricles. Ventricles are fluid-filled cavities inside the brain. I'm repeating again. Ventricles are fluid-filled cavities inside the brain. Okay? Now, the wall of those cavities are lined with the ependymal cells. So, these are the ependymal cells that line the wall of the ventricle, and if you see the ependymal cells, they have many cilia. You know that cilia are soft hair-like structures. Okay? Cilia are soft hair-like structures. So, in the ependymal cell, you will see cilia. Okay? So, ependymal cells are found, again, I'm repeating, ependymal cells are found in the wall of the ventricles. What are the ventricles? Ventricles are the fluid-filled cavities inside the brain. Okay? And you know that since the cavity is filled with fluid, you have fluid here, the cilia, when they move, they create wave in the fluid and let the fluid move. Not, when it is important that the fluid in the cavity always move. If it stays, without any movement, then the chance of getting infection is more. So it will move the fluid a lot, the cilia. Anyway, so if I ask you in the wall of the ventricles, what kind of neuroglial cells you will find? Ependymal cells. How you will know these are ependymal cells? They have cilia. Very good. Okay. Now we'll talk about the neuroglial cells. Those provide insulation. I have already talked about that. Insulation is important. So oligodendrocytes form insulation around the nerve fibers in the central nervous system. So if you see the insulation around the nerves in the central nervous system, nerve fibers, these are the nerve fibers, the yellow structure, and you see the insulation around the nerve fibers, the bluish color, that's the insulation. So this is the nerve fiber and the insulation is like this. Okay. So if you see the insulation is in the central nervous system, that is given by the oligodendrocyte. Okay. So now in the peripheral nervous system, the nerves are insulated by two types of cells, satellite cells and Schwann cells. So satellite cells will provide insulation around the cell body. So this is the neuron, you see this is the cell body and this is the process. So the satellite cells will insulate the cell body. So it is around the cell body. And Schwann cells will insulate the fibers, nerve fibers. But both are present in the peripheral nervous system, not in the central nervous system. Okay? Oligodendrocytes are in the central nervous system. So now you've got three neuroglial cells that provide insulation. One is oligodendrocyte, another is Schwann cell, and another is what? Satellite cell. Satellite and Schwann will provide insulation around the peripheral nerves. So remember those neuroglial cells, and just a couple of important information I have given from each type. You need to remember those, and I will give a few questions from there. Okay, so we are done with the supporting neuroglial cells. Now we'll talk about the main cell type, neurons. Okay? Now, neurons are different than other cells of the body. How they are different? Neurons are long-lived. The lifespan is very long. They can stay alive more than 100 years. 100 years or more, they can stay alive. You know that many cells of our body die, but the neurons can stay alive for 100 years or more. That is one important property. Number two, amitotic. A means absence, and mitosis is a type of cell division. Probably many of you know that. Mitosis is a type of cell division, and A means absence. That means the neurons don't divide no multiplication occurs in the neurons in general. There are a few exceptions, but in general we say that neurons are amitotic, they don't multiply. Now, once your nervous system is developed, the neurons will not multiply, will not produce new neurons. That's why you know that if brain stroke occurs, then the person, for example, the person got paralysis, the person will suffer from that in the rest of his life. Make sense? You have seen that many paralytic patients will not cure, they will stay like that. You can improve a little bit by increasing the muscle activities and like that. But the brain, why that happens? Because no new brain tissue is formed, no multiplication of brain cells occur. So the nervous system cells or neurons are amitotic. Yes.
So does that mean when you're born with all of your neurons that you'll...
No, I said that once it is developed. That
once
means
it's developed,
developed, the adult person,
okay.
then if the neurons die, no multiplication will go.
Okay.
Of course, in the early life that I have shown you, you have only few cells, then they multiply and move in different directions. So,
okay, so
during that time, they will multiply.
Okay.
Make sense? But once the nervous system is developed, then if the cells die, no new cells will replace them.
Okay.
Make sense?
Mm-hmm.
Now if I cut your skin, you know that very quickly, new skin is formed, so that happens in other parts of the body, but not in the nervous system. Okay. Depends on continuous supply of oxygen and glucose. Very sensitive. So you see two different things. One is neurons can stay alive 100 years or more unless you bother them. If you bother them, they will die very fast. Very quickly they will die. They are very sensitive, extremely sensitive. For example, if I stop the blood circulation to your hand for a few minutes, like this, when I leave the pressure, your hand will still stay alive. Now if I push here hard and stop blood circulation to your brain, how long you will stay alive? You will not stay alive. Right? You will fall and eventually will die if I keep stopping, even a minute or so. So you know that your brain tissue, neural tissue, needs continuous supply of oxygen and nutrition. If you stop that, they will die very fast. So two different things. If you don't bother them, they will stay alive longest, 100 years or more. If you bother them, they will die fastest, very quick. Okay? Because they need continuous supply of oxygen and glucose. Electrical signal. Neurons produce and transmit electrical signal. That's the function I have mentioned, that in one sentence, if I ask you, that's the function. So neurons are the only cells that can generate and transmit the electrical signal. So important property of neuron. Now, parts of a neuron. A neuron has a cell body and processes. So if you see the structure of the neuron, you have seen it before, the picture. So the neuron has a cell body. This is the cell body that has the nucleus and processes. Two types of processes arise from the cell body. From one side of the cell body, you see multiple processes, more than one usually, and those are short, multiple, and branched. So from one side of the cell body, a number of short and branched processes. You see branching. These are called dendrites. Okay? And from the opposite side of dendrites, a long single process arises, and that long single process is called an axon. So this is the axon. Okay. So in a typical neuron, remember I am talking about a typical neuron. Most common neuron has a cell body, multiple dendrites. How the dendrites look like? They are short and they have what? Branches. And a single long process that is called the axon. Now, just know that the cell body has a nice nucleus in the center, and in the cytoplasm you will see tiny granules. These dark or gray colored granules, these are called missile granules, also called missile bodies. Okay? So missile granules or missile bodies. These are gray to dark granules present in the cytoplasm of the cell body, the neuron. That is another important thing, so remember that. And let's see the picture of a neuron. This is a typical neuron, and you can see the cell body. These are missile bodies or granules. Okay, missile bodies, and these are dendrites. This is the axon. Now, the area where the axon arises from the cell body, that area is called Axon Hiller. So Axon Hiller is the area where the axon arises from the cell body, or we can say the axon gets attached to the cell body. Either way is fine. So that area is called the Axon Hiller. And you see that in this neuron, the axon is insulated by the Schwann cells. We know that in the peripheral nervous system, the fibers are insulated by Schwann cells. So these are the Schwann cells. Okay? They cover the axon and insulate it. So the nerve signal transmission will be very fast and efficient. Without loss of electricity, the signal will travel very fast. Okay? Another thing is that which way the signal, electrical signal will travel. You need to know that. The electrical signal will travel this way. Okay? Always. The electrical signal will travel in that direction. That means what? The dendrites will bring the signal into the cell body. So dendrites will do what? Bring the signal into the cell body. Okay? And cell body will process that signal, and then the signal will be taken out from the cell body by the axon. So axon will take the signal away from the cell body. Dendrites will bring into the cell body, and cell body will process that signal, and axon will take it out from the cell body. So that is unidirectional. Remember that all neurons will do that way. One way the signal will travel. Okay. Now, you see what happens. How the neurons are connected to each other. This is another neuron. Okay? Okay. So here, what happens, you see, the axon will give, this way the signal will go and will give signal to the dendrites of next neuron. And dendrites of next neuron will receive that signal and will bring again into the cell body. And then the signal will get up. Here you have another neuron. And these are the dendrites. Will receive the signal from the axon. Okay? So that's how the signal is transmitted from one neuron to the next. So dendrites will always receive the signal and bring into the cell body. This is important for you to know. Okay, myelin sheath. Myelin sheath is nothing but the insulation around the axon. And you have seen the insulation around the axon in that picture I have shown you here. That's the myelin sheath. Okay, that insulation. Okay, so nodes of Hiller. Another thing you need to know. You see, in between the Schwann cells, you have a small uncovered area. Those uncovered areas are called nodes of Hiller. So, the Schwann cells are not forming a continuous shape. There are gaps in between the Schwann cells. So those uncovered tiny areas are called nodes of Hiller. Okay? So from the neuron, you need to know those information I have given. If I ask you that, tell me which type of neuron is the most common, most abundant? Astrocytes. That's not the neuron, that's the neuroglial cells. Yeah. So a typical neuron, if I ask you, tell me a typical neuron has how many parts? It has a cell body and processes, right? What are the different processes? Dendrites and axon. And you know how they look like, right? Dendrites are usually multiple, they are short, and they have branches. Very good. And axon is what? Axon is single, long, and yeah. So dendrites do what? Dendrites bring the signal to the cell body, and axon takes it from the cell body. Away from the takes away from the cell body. Okay. And now, if I ask you what is the Axon Hiller? Yeah, where the cell body meets the axon, or axon arises from the cell body. Micelle granules. If I ask you what type of granules you will find, or bodies you will find in the cytoplasm of a neuron, those are the Micelle granules. Just know the name. Okay. So those are the important information about a typical neuron. Okay. Now, gray matter and white matter. In your nervous system, what happens? The neurons are arranged like this. These are the cell bodies, these are the axons. Okay? So, you see many neurons here. So, in your brain and your spinal cord, neurons are located like this. So, these are the axons, these are the cell bodies. And now, if you separate those two areas, inside the cell bodies, you have what? Those dark or gray micelle bodies. You know that. So, many micelle bodies here. And the axons are covered by myelin sheath. You know that all axons are covered with myelin sheath. Now, remember that the color of myelin sheath is white. White sheet. So these axons are covered with white myelin sheet, and in these cell bodies you have micelle granules. So when you see all cell bodies in one area and those cell bodies have micelle granules, it will look white or gray. Gray, right? Make sense? Because micelle granules are gray to dark color. Now, if you see all myelin sheet in one area, many myelin sheet in one area, and they have white covering myelin sheet. Many fibers are in one area, axons, and they are covered with white myelin sheet. So this area will look what? White. This area will look gray. That's why we see different colors in the brain and the spinal cord. If you cut the spinal cord and the brain, you will see some areas look gray. Those are gray matter. Some areas look whitish. Those are white matter. So, and now you know why gray matters look gray and white matter area looks white. Make sense? Okay.
So when someone has like a serious brain injury and they say that there's gray matter, is that what that means?
Yes, definitely. But remember that if this is your brain, if you cut the brain, the outer part is gray matter, and the inner part, this is a piece of brain. Okay, so this is the outer surface of the brain. If you cut this part, outer part is the gray matter, and inner part is the white matter. So if you get an injury from outside, then which part will be affected? Gray matter, because that's the outer covering, outer part of the brain. So in the outer part of the brain, you have gray matter. So if you get injury from outside, the gray matter will be affected. Okay, so if I ask you in which area you will find many axons or fibers?
That's white.
White matter. Why? White matter looks white because the myelin sheet, the fibers are covered with myelin sheet, right? In which area you will find many cell bodies? Gray matter. Okay, so that's the difference. Now, another thing you need to know. Inside the brain, you will also find tracts and nuclei. So inside the brain, you will find gray matter, white matter, tracts and nuclei. Those are the commonly seen structures inside the brain. Now you already know the gray matter and white matter. Now I'll tell you what is a tract and a nucleus. In certain areas of your central nervous system, you will see the fibers are heavily, tightly packed. Many fibers are bundled tightly. So those are called tracts. The difference between white matter and a tract is in white matter you see many fibers, but they are not bundled tightly, but many fibers in white matter. In a tract, you will see many fibers, but they are very tightly bundled. So those are tracts. Nuclei. In the nuclei, you will see many cell bodies are heavily packed, clustered. So that's the nucleus. The difference between the gray matter and a nucleus, nuclei is plural. So in the gray matter, you will see the cell bodies, many cell bodies, but they are not heavily, clustered, like side by side. In the nuclei, you will find many cell bodies are heavily packed. So that's the difference. Okay. So we see in some areas in our brain and the spinal cord, the nuclei are present. That means the cell bodies are heavily packed together. Okay. Now we'll talk about different types of neurons. We have talked about different parts of a neuron, but we did not talk about different types of neuron. Neurons, three types of neurons we see in the nervous system. What are those? Multipolar, bipolar, and unipolar. The neuron I have shown you a few minutes ago, that's the typical neuron. When I said this is a neuron, what type of neuron? Typical. That means that's the most commonly seen neuron. And that is the multipolar. So I am drawing that again. So I showed you this neuron before. So this is a multipolar. Most common type, remember, most common type neuron is multipolar. That means it has a cell body, multiple dendrites, and a single axon. That's the typical neuron. Now, bipolar. If you see a bipolar neuron, it has one dendrite, single dendrite, and a single axon. So how many total processes? Two. One axon and one dendrite. So that's why it is called bipolar. Bi means two. Okay. Another type of neuron you will see. This is the cell body of the neuron, and the process is like this. That means the cell body is attached to the side of the process, not to the end. And this is a single process, only one process, and cell body is attached to the side of the process. So if you look, sometimes you may think that it has two processes, but actually it is a single process. Okay? So one process, so this is unipolar. Okay? So if you see a single process, that's the unipolar. If you see one axon and one dendrite, that means how many? Two processes, that's the bipolar. If you see one axon and multiple dendrites, that's the multipolar. Okay? And multipolar is the most common or typical neuron. Here in this picture, you can see those three types of neurons. You see both are multipolar, although they look different from each other. This is the cell body, and these are the dendrites, and one axon. Now, always look when you see how many. If I ask you, tell me how many dendrites. See how many arising from the cell body. So here I'll say two dendrites. Here I'll say 1, 2, 3, 4, 5, 6. I won't count these branches, right? Those are the branches. So always, for example, here, how many dendrites? You have 1, 2, 3. These are not the, we won't count the branches. Like if I ask you, show you a tree, like there are a few trees, and ask you how many trees are there, you won't count the branches, right? You will only count that how many stems are now coming out from the ground. Okay. The reason I said that, because you see this is a bipolar. This is also a bipolar. Now from the cell body, only one dendrite and one axon. That means two. Don't count this. If you count this, then you will say it is multipolar, but it is not. You have to count it. And this is a unipolar. So this is only one process. That's the axon. Okay? Single axon, and cell body is attached to the side. So just know that I have mentioned. Synapse. synapse indicates connection. Neuron to neuron or neuron to muscle. Both connections are called synapse. Neuron to muscle, you have seen that is called neuromuscular junction. When we talked about the muscle tissue, I have shown you a neuromuscular junction. That means this is the axon terminal and this is the muscle fiber. That's the neuromuscular junction. We have talked about the structure of a neuromuscular junction in detail. So that is one kind of synapse where the nerves are talking to the muscles. That's the neuromuscular junction, one type of synapse. Another type of synapse between the neurons. That also I have shown you when I did draw here how neurons are connected to each other. You remember that? I showed that next neuron is here and these are the dendrites. So this is the another type of synapse. That's neuron to neuron synapse. This neuron is talking to the next neuron here, the axon terminal and dendrite. Okay? So two types of synapse we see in our body. Nerve to the muscle fiber and neuron to neuron. Okay? Reflex. The definition of reflex is a rapid autonomic response to stimuli in which a particular stimulus always causes the same response. This is a nice definition. I like it. What is that? If your body responds to a stimulus same way, if I present the stimulus again and again, that is the reflex. For example, if I ask you to open your eye and I put very powerful flashlight on your eye, suddenly your pupil will do what? Constrict, right? You know that pupil will constrict. Now, if I do it again, your pupil will constrict again. If I do it again, your pupil will constrict again. Okay? So that is the reflex. It will respond same way again and again by repeated stimuli. Okay? So that's the reflex. If it just responds once and then it doesn't respond next time, that's not a reflex. Okay? That's why we say autonomic. You cannot stop. You cannot say that I will not constrict my pupil. It will happen. You have no control. Now, a reflex to occur, you need some components that will complete the circuit. What is that? For example, you know that another example of a reflex is when you hit the patellar ligament, your leg shoots forward. Okay? So, first component here for this reflex is receptor. You have receptors in the patellar ligament, and when you hit, those receptors will be activated and will send signal to your spinal cord. So that signal will go to the spinal cord. So first the receptors will receive that signal, and then sensory neurons will take that signal to the spinal cord, because we know that signal goes to the central nervous system, that means towards the central nervous system by sensory nerves. Sensory nerves take the signal at the very beginning, I said, towards the central nervous system. So this signal is going towards the central nervous system, that means it is sensory. Then you have the integration center. In this case, your spinal cord is the integration center. That's the center part of the central nervous system. You know that central nervous system is the integration center, where it will process that signal and will send a signal back to the muscle, leg muscles. So who will take the signal from the spinal cord to the leg muscles? That's the motor, because we know that motor will take the signal away from the central nervous system towards the muscle. And last component is the effector. In this case, the effector is the muscle, because muscle will perform the action. Okay? We move the leg. So those are the 5 components of your reflex arc. If you block any of those components, then the reflex will not be completed. Reflex will not occur. For example, here is the patellar ligament. So you have receptors here, and the signal is going to the spinal cord. This is your spinal cord, and then coming back to the muscle. Okay? So receptors, then signal is going to the central nervous system, to the spinal cord. That's the sensory neurons, and then integration center is the spinal cord, and from here the signal is going to the muscle. This is motor, and effector is the muscle. Okay, effector that will perform the action. That's the effector. Now, if you block anywhere, for example, if you destroy the sensory nerves, neurons, then the signal will not reach here. The reflex will not occur. The muscle will not be activated. If you block here or if you destroy here, then the reflex will not be completed. So to complete a reflex or to a reflex occur, you need the reflex arc. Okay? And that's why we do those testing. Why we do the knee jerk testing. We see if the person's nervous system is working. Everywhere it's working fine. Okay? So this is one test that the spinal cord is intact and the signal is coming back to the spinal cord. Okay? So that's the reflex and the reflex arc. So just remember, if I ask you what are the components of a reflex arc, those are the 5 components. You now understood that why those 5 components are essential. Okay? Let's take a 5-minute break and come back quick so we can finish. Nervous system is very interesting but very complicated too. So if you understand, you will enjoy it.
You have to do lots of brain research for education.
Second?
You have to, in education, you have to do lots of brain function.
Yeah, I spent whole of my life in doing neuroscience research. My field is neuroscience actually. That's why I feel nervous when I teach neuroscience at this level. I teach another class at WashU. That's higher level neuroscience. So it is difficult for me to stop, and only stop and talk about the basic things like here at this level, yeah. But yeah, I think those things I have mentioned, those are very important things. Yes.
So, it says attachment. Did we need to know the attachments for these 3?
Yeah.
Okay.
Those are
And
the
it's just the
muscles.
humerus and the scapula, right? They all attach for the humerus and the scapula?
They do, because they move the shoulder joints. Okay.
Right.
Now, when I say attachment, just where they are located. Okay, so those are the locations.
Okay.
Not the origin insertion.
Okay.
Okay.
With like pages from the book. She did some sort of program through I think there's other people, but there's one or two that she's I know it took me like 2 hours or something. My mom helped me, but I still need to like do the underside on the That's why I'm doing this. I'm just like, it's way too expensive.
Yeah.
Okay, so we'll start. Some of the students are still outside. Let me start, and they will come. Yeah, they will, yeah, you can call them. Okay, so you know that the nervous system is divided into two divisions, the central nervous system and the peripheral nervous system. Now we'll talk about the central nervous system. Central nervous system has two structures, the brain and the spinal cord. So now first we'll talk about the brain. Today we'll talk about the brain. In next class we'll talk about the spinal cord. Okay. So in this part we'll talk about the protection of the brain. Then the ventricles of the brain. You remember today I said that inside the brain you have fluid-filled cavities, and those fluid-filled cavities are called the ventricles. Okay? So, what are the ventricles present inside the brain? We'll see that. Okay? And the fluid that fills the ventricle, that fluid is called cerebrospinal fluid, or CSF. Cerebrospinal fluid, or CSF. So we'll see how the CSF comes inside the ventricle. to the ventricle, from where the CSF comes. We have already talked about white and gray matters. We'll see in pictures how they look like and where they are located in the brain and the spinal cord. Then we'll talk about the functional areas within the cerebral cortex. In the cerebral cortex of the brain, you have different functional areas. What does mean the functional areas? For example, very simple. When you see something, you have a particular area in your brain, in the back, in the occipital lobe, here. That will receive that visual signal and will process that, not the whole brain. That's not good, right? If your whole brain processes everything, it will be chaotic. So, your visual signal will go to the visual part of the cortex. When you listen something, that auditory signal will go to your temporal lobe. A particular part of the temporal lobe will receive that signal. Your smell will go to another area. So, in the cerebral cortex, you have different functional areas to process different functions. So, we'll see the important functional areas in the brain. Okay? What are the important functional areas? Only we'll see the important few areas. Then we'll talk about homunculi. Homunculi is your body map in the brain. That means what? The whole body map is present in the brain. So we'll see which body maps are present in the brain and where those body maps are present in the brain. Okay? So those body maps are called homunculi. Okay? So we'll see that. That's interesting. Protection of the brain. Which structures protect your brain? It's a common sense, you know some structures already that structures protect your brain. Your brain is the most important and delicate organ of the body. Not only important, it is the most, sensitive and delicate organ. So, it must be heavily protected in our body. And that's why knowing the protection of the brain is important. So, brain is heavily protected by a number of structures. You know that brain is located inside the cranial, inside the skull. And you have the cranial cavity inside the skull, your brain is located there. So bones, cranial bones protecting your brain. Then brain is covered by meninges. Meninges are connective tissue membranes. 3 different meninges or 3 layers of connective tissue membrane are present around the brain. So we will see what are those 3 connective tissue membranes around the brain. Then watery cushion provided by the cerebrospinal fluid. In between 2 of those 3 membranes, you have cerebrospinal fluid. So fluid is located in between 2 meninges, and that fluid also protects the brain. It provides cushion. Blood-brain barrier. This is a different type of protection. Blood-brain barrier is nothing but the wall of the capillaries, blood capillaries in the brain. So write it down. Blood-brain barrier is nothing but the wall of the blood capillaries in your brain. Now the question is, how the wall of blood capillaries provide protection to the brain? This is interesting. You know that inside your brain you have capillaries those are the tiniest blood vessels, capillaries, are present. Everywhere in the body we have capillaries. Now, the wall of brain capillaries are made differently than the other capillaries of the body. So, I am repeating again, the wall of brain capillaries are made differently than the wall of other capillaries of the body. In the wall of the capillary, you have what? If you see under the microscope, the wall of the capillary, this is the wall of the capillary, you have many holes or pores everywhere in the body. In the wall of the capillary, you have pores, tiny holes. Now, if this is the wall of a capillary in other parts of the body, now we'll see the wall of the capillary in your brain. In your brain capillary wall, you have the holes, but those holes are much smaller than the holes present in the capillary wall of other parts of the body. Okay, so you can now easily think that through which capillary wall the chemicals will easily get out. Through this one or through this one? This one, right? Because the holes are bigger. So from the blood, many chemicals will easily get out and will enter into the tissue. That's fine in other parts of the body. But if that happens in your brain, if many chemicals can easily get out and enter into the brain tissue, then that can cause harm to your brain tissue. Because in your blood, you have some harmful chemicals too, you know that. So if they can easily get out into your brain, will kill the neurons, because neurons are very sensitive. That's why the wall of the capillaries in your brain are made different. The walls are made different. And that is the blood-brain barrier. And that protects your brain from chemical harm. It will not let all chemicals to enter into the brain from the blood. And you know that very often we take the medicines, right? We take the medicines for many different problems. Now, if all those chemicals can easily enter into your brain tissue, they will cause harm. That's why the chemicals, most of the medicines or chemicals that you take cannot cross the blood-brain barrier, cannot cross the wall of the blood capillaries in the brain, and cannot enter into the brain tissue. Okay, so that's the blood-brain barrier. So other structures, bones, membranes, and watery cushion, they provide mechanical protection, I can say. Mechanical protection, right? If something hits from outside, those things will prevent or protect your brain. Now, the blood-brain barrier is providing chemical protection from the chemicals. Protection from the chemicals. Just write it down. Okay, now you see here the structures, those are present outside of the brain and protects the brain. We'll start from outside to inside. Above the skull bones, you have other structures you already know. Skin of the skull, right? That is the first layer. Then under that, you have periosteum. Periosteum is the covering of the bone. You know that. So, skin of the skull. Then next is the periosteum of the bone. Then you will hit the bone, skull bone. Then under the bone, as I have mentioned, you have 3 connective tissue layers or membranes. What are those? Outermost one is the dura mater. Those are the meninges. outermost one is the dura mater, then middle one is the arachnoid mater, and innermost one is the pia mater. So those three are the connective tissue membranes called the meninges. Okay, those are called meninges. Now, dura mater is the outermost one and the toughest one. If I ask you which one is the outermost one, which one? Dura. And that is the toughest one. Okay. So if I ask you which one is the toughest one, that's the dura mater too. Then next layer or membrane is called the arachnoid mater. And under the arachnoid mater, that space is called subarachnoid space. Makes sense? Sub means below, right? So under the arachnoid mater, the space is called subarachnoid space. In that space, you have the cerebrospinal fluid, or CSF. Okay? So if I ask you the CSF is present in which space, that's the subarachnoid space. And that fluid provides protection. And then the innermost membrane is the pia mater. Pia mater is the thinnest one. Thin, very thin, extremely thin. And since this is the innermost one, this membrane is directly attached to the surface of the brain. This is the innermost one, pia mater, and is directly attached to the surface of the brain. Okay? You cannot separate that actually from the brain. So those are the three meninges. Okay? They protect the brain as well as the cerebrospinal fluid in the subarachnoid space. Now we will go to the next slide. So we have talked about three meninges. Ventricles. I have already mentioned 5 times what is a ventricle, right? Ventricle is a fluid-filled cavity inside the brain. And fluid is the cerebrospinal fluid. And you know that in the wall of the ventricles we have the ependymal cells, one type of neuroglial cells, those have cilia. Okay? What are the ventricles present in the brain? In the brain, you have 2 lateral ventricles, one in each half of the brain. So we have 2 lateral ventricles. And we have 1/3 and 1/4 ventricle. These are the name of the ventricles: lateral ventricle, third ventricle, fourth ventricle. How many lateral we have? 2. How many third ventricle we have? 1. And how many fourth? We have 1. Okay, so total 4 ventricles. And those ventricles are connected to each other. They are not separated. They are connected to each other. So the fluid can flow from one ventricle to the next. Okay? Since they are connected. So here in this picture you see, although the ventricles are located inside the brain, so you won't see the ventricles from outside. But they have shown the position of those ventricles inside the brain. If you see the brain from the front and try to see where the ventricles are located inside the brain, they are located like this. These two are lateral ventricles. This is the third ventricle, and this is the fourth ventricle. Okay. So lateral, third, and fourth. Now you see that the lateral ventricles are connected to the third ventricle. How they are connected to the third ventricle? By interventricular foramen. So the fluid can move from the lateral ventricles to the third ventricle through the interventricular foramen. Third and fourth ventricles are connected by cerebral aqueduct. Cerebral aqueduct is a narrow tube-like structure that connects the third and fourth. So this is the cerebral aqueduct. So if I ask you how many ventricles you have in the brain, total 4. What are those? 2 lateral, 1 third, 1 fourth. How they are connected? Lateral ventricles are connected to the third ventricle by interventricular foramen. Third ventricle is connected to the fourth by cerebral aqueduct. So remember those information. Okay. Now, why the ventricles are connected to each other? They are connected to each other because if the fluid pressure, the pressure of fluid increases in any of those ventricles, then what can happen? The fluid can move to the next ventricle and reduce that pressure. If they are not connected, then the fluid pressure will increase and will cause pressure on the brain tissue. So since they are connected to each other, if pressure increases in one ventricle, then it can be reduced by movement of the fluid from one ventricle to the next one. Okay. Now, there is a clinical condition that is called hydrocephalus. Have you heard that, anybody? Hydrocephalus. So what happens in hydrocephalus? What causes hydrocephalus? If, for example, a tumor in your brain tissue grows here, or ependymal cells also can cause tumor, multiplication of ependymal cells. Anyway, so if a tumor is formed here, then the fluid will start to accumulate inside this ventricle. Okay? Or if the tumor is here, then fluid will accumulate in this area, and the ventricles will get bigger because of the fluid, increased amount of fluid. And when the ventricles will get big, your whole brain will get bigger, will expand. Now, hydrocephalus is seen commonly more in children than adults. You will see the head of a child gets bigger in hydrocephalus. The reason is, it doesn't mean that hydrocephalus only occurs in children. The hydrocephalus big head is seen in children. The reason is, in early life, very early life, the cranial bones are not fused to each other. In very early life, the cranial bones are still not fused. So you have a lot of space in between the cranial bones. They normally, this is one cranial bone, stay like overlap like this, and there is space. So now, when the brain grows, since the cranial bones are not articulated strongly, they can move apart. They can move away, and that's why you see the head is big in children. In adults, since you know that sutures heavily connect the cranial bones strongly, so the skull bones cannot move away from each other. That is more dangerous. When pressure increases in your brain, if your bones can move away, it will reduce the pressure of the brain. Now, if the bones cannot move, it's still there, but your brain is trying to expand, that will cause even more problem and damage to the brain, because your bones are not moving, your brain is expanding, not getting more space. Okay? So that is more problematic if it happens in adults. Yes?
Would you mind writing it on the board, see how it's spelled?
Hydrocephalus. Hydro means what?
Water.
Water. Cephalic. Cephalic means the brain, upper part, you know that cephalic end, right? And caudal end. So cephalic is the brain or head part. Cephalic indicates the head part, and hydro is fluid or water. So water or fluid in the head. That's the hydrocephalus. Okay, the functions of the cerebrospinal fluid. Number one is protection. You have cerebrospinal fluid in the subarachnoid space, you know that, around your brain. So you know that we move our head a lot, right? We move our head in different directions a lot. Particularly when you go to Six Flags. I think probably all of you have gone to Six Flags. And you didn't go? Never?
No, I've been, I always get a motion sickness.
Oh, yes. Okay. So when you ride on roller coasters, you know that how fast the roller coasters move from one direction to suddenly, right? So your head is moving suddenly from one direction to another. If you didn't have any cerebrospinal fluid around your brain, the subarachnoid space, then your brain would have hit the hard skull every time you are suddenly moving your head. That fluid is preventing that to happen. Okay, so cerebrospinal fluid is very important cushion. It protects the brain. Another function is cerebrospinal fluid contains nutrients. Okay, and that nutrients keep the brain healthy. Keep the brain healthy. And some chemicals are also present in the cerebrospinal fluid and helps in chemical signal transmission. But two functions are really important here. One is protection, another is giving nutrition to the brain and keeping it healthy. Okay. Now, one thing we need to know that from where the cerebrospinal fluid comes into the ventricles. If you see inside the ventricles, this is the lateral ventricle, you will see choroid plexuses. Choroid capillary plexus, name choroid plexus. Okay, choroid plexus is nothing but capillary plexus attached to the wall of the ventricles, and the cerebrospinal fluid is secreted from the choroid plexus. That's how the cerebrospinal fluid comes into the ventricle. Okay, secreted by the choroid plexus, and choroid plexus is attached to the wall of the ventricle. Which wall? Choroid plexus is attached to the roof of the ventricle. So ventricle is a cavity, so it has a roof, and the choroid plexus, that capillary plexus, stay attached to the roof of the ventricles. Let me mention here. Yeah. Okay, so that's all about ventricles. We need to know how many ventricles we have, what are the ventricles, fluid-filled cavities, and how they are connected, right? Those are the things. And who secretes the fluid into the ventricles. What is hydrocephalus? I may also ask. Okay. Now we'll see the parts of an adult brain. We divide the brain into 4 parts. We divide the whole brain into 4 parts. The largest structures of the brain are cerebral hemispheres. Hemi means what? Say loud. Half. So sphere. Sphere means whole round structure, right? So half of that whole sphere is hemisphere. So 2 hemispheres we have in the brain, and those are the largest parts of the brain. They join together, 2 halves, and form the cerebrum. Okay? So whole cerebrum has 2 hemispheres. Those are the largest. If you see the brain at a glance, you will see the cerebral hemispheres because those are the largest parts. Then you have diencephalon. Diencephalon has 3 structures in it. So diencephalon is the next part. It has 3 structures: thalamus, hypothalamus, and the pituitary gland. You know that that's the master gland, right? Where it is located. So thalamus, hypothalamus, and pituitary gland, those are the 3 structures in the diencephalon. Or we can say those 3 structures together called the diencephalon. Then the brain stem. Brain stem has 3 structures. From the top, if you go down, midbrain, then pons, and the medulla oblongata. So midbrain, pons, and medulla oblongata, those are the 3 parts of the brain stem. Okay? And last, in the back of the brain, you have another structure that is called cerebellum. Okay, cerebellum. So those are the 4 parts of the brain. Here, if you see the brain, the largest, this is, you are looking at the brain from the side. So you see 1 hemisphere. Another one is in the other side, right? So the largest part, that whitish structure, that's the hemisphere. Then the diencephalon is the part that has 3 structures in it. They didn't show those 3 structures, but let me show you here. This is the diencephalon. Now, if you draw, when you do the dissection and see the brain in the model, it will be easy if you just listen. If you draw an egg, you know the egg, chicken egg, like this here. This part is called the thalamus. So this is the thalamus. There is no boundary that you can see. So you have to imagine that this is an egg and this is the thalamus. And below the thalamus, this part is called hypothalamus. Hypo means below. Okay? And so this is the hypothalamus, and this is the pituitary gland. Okay? So those are the 3 parts of diencephalon. And then the green structure is the brain stem. From the top, you see midbrain, pons, and medulla oblongate. And in the back of the brain, you see the cerebellum. Okay. So those are the 4 parts. Now I will briefly tell you the function of those 4 parts. Just write it down. Okay. Cerebral hemispheres are the structures where many functional areas are located. You remember I have mentioned before what are the functional areas. In certain areas of your brain, certain functions are processed. For example, when you see something, the visual signal goes to the back of the cerebral hemisphere. When you hear something, that auditory signal will go to your temporal side of the brain. So, on the cerebral hemisphere, in different locations, you have different functional areas. So, we can say that functional areas are located in the cerebral hemispheres. That's the function. Then the diencephalon. I will mention one or two functions from each part of diencephalon. First, thalamus. Thalamus is called the major sensory relay station. Major sensory relay station. Almost all sensory signals must be relayed in the thalamus. Your visual signal, your auditory, those are sensory. Your pain, touch, temperature, all those sensory signals are relayed in the thalamus. That's why we say this is the major sensory relay station. Just in one sentence, that's the function of the thalamus. Hypothalamus performs a number of important functions. I'll just mention 2 very important functions of hypothalamus. One is temperature regulation. Regulation of body temperature. You already know this. Hypothalamus controls the sweat glands. Number 2, it's a very interesting function. Food and water intake. How much you will eat and drink. That's the food and water intake. That is controlled by your hypothalamus. That's interesting. Like when you get thirsty, if I give you water, you will drink very fast. Then if I give you more and more, what will happen? Gradually, you will lose the interest, right? Then you will say, no, I can't. Even if I ask you to drink, you say, I won't drink anymore. I am done. So your hypothalamus will inhibit your desire to drink. Okay. Same thing happens when you eat. You start to eat, and you think, I will eat everything, and you are hungry. Then what happens? You gradually lose the interest, right? And you stop eating. So those are controlled by your hypothalamus. There is an interesting experiment on hypothalamus. You can Google and find it in the internet. I'm sure it should be there. 2 groups of mice were taken. One group of mice, what they did, they let the mice drink and eat as they want. Okay? So, as much as they can. So, then they destroyed the hypothalamus in one group of mice, and in another group of mice, the hypothalamus was intact. They did not touch. And then after a couple of months, they found that the group of mice had destroyed hypothalamus. They got like huge, like few times more, bigger, like giant, than the other group, because they lost their control where to stop eating and drink. So they continuously ate. In humans, sometimes that happens, hypothalamus disorder that can cause the person, or make the person obese and huge. Those people cannot stop eating or drinking; they will keep eating or drinking. So it could be related to hypothalamus. Okay. Okay. So those are just remember those two functions. Pituitary gland is called the master gland. You already know that. That means what? It is the gland that controls the other endocrine glands of the body. That's why it is called the master gland. Okay. In the brain stem, just know that important control centers are located. Which important control centers are located in the brain stem? Your cardiac centers, your respiratory centers. Cardiac centers control the heart. Respiratory centers control your respiration or breathing. So those centers are located in the brain stem. Okay. So in the brain stem, you have those important control centers. Which control centers? Cardiac and respiratory. Cardiac will control what? Heart, and respiratory will control your respiration or breathing. Now if you destroy the brain stem, your heart will stop, your respiratory centers will be affected and you will stop breathing. So what will happen? You'll die if those centers are destroyed. Okay, now cerebral hemispheres are the largest structures of the brain, and in each hemisphere you have two hemispheres. In each hemisphere you have 4 lobes. So here you see in the bottom part it says that 5 lobes. Just cross out the insula. Yeah. So each cerebral hemisphere has 4 lobes, and those lobes are named after the bones. Those are located above the part. For example, frontal lobe is under the frontal bone. Temporal lobe is under the temporal bone. Parietal lobe is under the parietal bone, and occipital lobe is under the occipital bone. In general, we say that those bones overlie those areas. So frontal lobe, parietal lobe, occipital lobe, and temporal lobe. Okay, now if you see the surface of the cerebral hemisphere from outside, you will see there are many shallow and deep grooves are present on the surface. Many shallow and deep grooves. Grooves are, how to say that? Grooves are depressions, like this. Depressions. So many shallow and deep grooves. Deep grooves are called fissures. So deep grooves are called what? Fissures. And shallow grooves are called sulci. S-U-L-C-I, sulci. Some people say sulci, that's fine. I love to say sulci. If you want to say sulci, that's fine. Okay, so fissures and sulci. So if I ask you which groups are sulci, those are shallow grooves. Okay, if you see the groove is deep, that's the fissure. So if I draw here, you are looking from the side. This is a groove, and this is another groove. Okay, and this is another groove, and another groove. So this is a sulcus. Sulcus is one. When we say more than one, we use sulci. Okay, sulci is more than one. So this is a sulcus, and this is a fissure. Okay. What is this then? It's a loud sulcus, right? And this is
That's a fissure.
another sulcus.
Oh.
Fissure is
I saw
deep,
the light
right?
going out, right.
This is what?
That's a fissure.
Fissure. Fissure is deep, and sulcus is shallow. Now, in between the sulci, this area, you see this area, this is called gyrus. Gyri is plural. If more than one, we say gyri. Some people say gyri, that's fine. So gyri are the areas between the sulci. Okay, so brain area between two sulci is the gyrus. Okay. So this is one sulcus, this is another sulcus, and this is the gyrus. Okay. So you will see those structures a lot on the surface of the cerebral hemisphere. You see here, if you see the cerebral hemisphere, it has 4 lobes by 4 different colors. And you see many gyri in between sulci. Now, if you cut the cerebral hemisphere and take a small chunk out from it and see the outer part of the hemisphere is the gray matter that I showed you before. Outer part is the gray matter where you see many cell bodies, and inner part is white. That's the white matter. So you see here, if you take a piece of cerebral hemisphere out, this is the outer part, and this is the gray matter, and this is the inner part, that's the white matter. Okay. So if something hits from outside, first the gray matter will be destroyed. Now, you see here, these are the grooves, but this one is shallow, this one is very deep. So this is the sulcus, this one is the fissure. Okay. Going in deep. Okay. So cerebral hemisphere has many functional areas. We'll just see few important functional areas where they are located. Okay. This is important both for your lab and the lecture. This is the back of the brain, back of the cerebral hemisphere. This is the front. So, in the very back part of the cerebral hemisphere, you have the visual area. That is called the primary visual cortex. So, your visual signal, when you see something, that signal from the eye will go to that area and will be processed there. Okay? That's why we say that primary visual cortex. This is the temporal lobe here, and this is the temporal lobe, and the lobe, you have an area, small area, that is called primary auditory. That means the sound signal that you hear goes there and is processed there. So that area is the primary auditory cortex. Auditory is the audition, hearing. Okay. Then this sulcus, it is a prominent sulcus. This is called central sulcus. Why it is important? It separates the frontal. This whole lobe is the frontal lobe. Okay? And behind the central sulcus is the parietal lobe. So the central sulcus, just write it down. Central sulcus separates frontal and the parietal lobes. Those two lobes are separated by the central sulcus. Okay? Now, you know that this is a gyrus and this is another gyrus because these are sulci. So in between two sulci, this is a gyrus. In between these two sulci, this is another gyrus. Now, the gyrus located in front of the central sulcus, that is this one, this is called primary motor cortex. So primary motor cortex is the gyrus in front of the central sulcus. And primary somatosensory cortex is the gyrus behind the central sulcus. So this is important, okay? These two areas, central sulcus. In front of the central sulcus, you have primary motor. Behind the central sulcus, you have primary somatosensory. Okay? So motor is related to the movement. So this area controls the movement. Just know that since it is motor, primary motor, this is the main area in the brain that controls the movement of the body parts. Primary somatosensory. Somatosensory means pain, touch, temperature. Those sensations you feel in your skin. So just you can write it down that somatosensory includes pain, touch, temperature. Those you feel in your skin. And those signals are processed in primary somatosensory cortex. Another area is the Broca's area. Try to find where the Broca's area is. Broca's area is in the frontal lobe. Broca's area is in the frontal lobe. Broca's area is the speech area. Speech, when you talk. That production of speech is controlled by the Broca's area. So Broca's area helps in the production of speech. Broca's is the name of a scientist, French scientist. He was a neurologist too. He had a patient who had problem in speech, severe difficulty in speech. So he was a patient for a long time, Dr. Broca's patient for a long time. Then when that patient died, he took his brain out and studied under the microscope, and he found that all other areas were intact, but only the Broca's area was not intact. There was some destruction in the Broca's area. So Broca's was the first scientist or neurologist who indicated that this area is responsible for the production of speech because his patient has had difficulty in production of speech. Then other scientists later confirmed that, and this area was then named as the Broca's area. Okay? So remember, if I ask you, Broca's area is responsible for what? The production of speech. Okay? Good. Whole frontal lobe is responsible for the higher brain functions, higher order brain functions. So now, what are the higher order brain functions? Just think this way, it will be easier. Those functions of our brain separates us from other creatures. How our brain is different than other creatures. Many things all of us we do. For example, urination, defecation, those are lower brain functions. All of us we do. All creatures, other creatures also do that. Higher brain functions are your intelligence, your strong emotion, your judgment, the ability to judge. Okay? So those are higher brain functions, and those are controlled by the frontal lobe. That's why the frontal lobe is much bigger in human than other creatures. In other creatures, although in big creatures you will see the brain is huge, but the frontal lobe is small. In human, the frontal lobe is pretty big. So just remember where those areas are located, number one, and what they do. Only those areas I have mentioned. Okay? Here, in your brain, you have seen motor area, primary motor area, you have seen primary somatosensory area. So some areas are motor areas, some areas are sensory areas. We know that. There are some other areas present in your brain. What those areas do? They combine two or more functions, two or more different functions together. So those areas are called association areas. Its name is telling you what is that? Association. Association areas combine or associate two or more different types of functions. For example, when I am talking, when I talk, you do what? You see, you look at me, so your visual signal is going to your visual cortex. At the same time, you are listening. So your auditory signal is going to the auditory cortex, right? And you know that I have shown you that visual in the back and auditory is in the side here, far away from each other. There is another area in your brain where those two signals are combined together. That's the association area. Now, why that is important? Why that combining is important? Just think that when you are looking at me and I am moving my mouth like this, sound is getting out. Now, if you see that I am moving my mouth, but sound is coming from the skull, would it make sense? What will happen? You will get puzzled, right? You will get confused. That's not right. Something is wrong here, right? Why the sound is coming from the skull? Okay, so you see that when you see and hear, two things must be combined to make sense that yes, they're linked to each other. And that's why you need a third area where two or more modalities or functions should be put or combined together. Those are the association areas. Okay. So, the location of white and gray areas, gray matter, in the brain, in the cerebral hemisphere, you have seen this before. Outer is the gray matter, inner is the white matter. Now, if you see the brain stem and the spinal cord, you will see the gray matter is located in the inner part, and the outer part has the white matter. So, in a sense, we can say it is opposite in the brain stem and the spinal cord. The white matter is the outer part, and gray matter is located inside. Okay? So, if I ask you that in the brain, white matter is where? Inside, right? In the spinal cord, white matter is in the outer part. Just know this, that a structure that is called the corpus callosum that connects two hemispheres together. Two hemispheres, we have two hemispheres, they are connected to each other by a structure, that white structure you see, that is called the corpus callosum. Okay? So if I ask you, you should be able to answer that. Okay. Homunculi. You remember I said that in your brain you have whole body map. In certain areas in your brain, you have the whole body map is present. In two areas, your whole body map is represented or present. One area is your primary motor cortex, and another area is the primary somatosensory cortex. You already know those areas. You see here. In front of the central sulcus, you have primary motor cortex, and behind the central sulcus, you have the primary somatosensory cortex. So, your whole body map is presented here as well as here. So, you have two homunculi, two maps of whole body in the brain. One in the primary motor cortex and another in the primary somatosensory cortex. So, we have sensory homunculus and motor homunculus. Culi is plural. But the problem is, what problem? You see whole body is represented there, but what's the problem here? Anybody? Can you notice any problem? Feel free to see any problem in that map. Is all parts are correctly represented or not?
They're like larger and smaller.
Very good. Yes. Say loud. Everybody will listen.
They're like disproportional, like larger.
Exactly. So, homunculi are disproportionate body maps. Now I have added this new word that homunculi are body maps in the brain. But what kind of body map? Disproportionate map of the body. What does it mean? Some areas are represented in larger parts of the brain, and some larger areas are represented in smaller areas in the brain. Okay? That means, again, some larger structures are occupying smaller areas, and some smaller areas of the body are occupying larger or bigger areas in the brain. That's why we say disproportional. Now, if you see this guy will look like that big head, big face, head part is big, okay? Trunk part is small, hands are big, and feet are big, but the arm, forearm, those are smaller. So, but the hands are very big, large. So they will look like dwarf. Homunculus means dwarf, dwarf person. The term homunculus means dwarf person. Dwarf person, their trunk is small, but head part is big, look like. So that's why it's a homunculus. The last thing, any idea why you see the hands or fingers are very big? Common sense. Yes, we manipulate. We use the fingers and hands a lot, right? So to touch something, to feel something, we always use the hands. So in both sensory and motor areas, those structures are largely represented in larger area because we touch or feel, that's the sensory, and also move the fingers to hold something or always move the fingers much more than other parts. That's why those areas occupy bigger parts in the brain. So that's why it is disproportionate. And interestingly, if you use one area more than other areas, this map is not fixed. This map can be changed. That is called the brain plasticity. I did many research on that. One experiment, this is a nice experiment. If you try to see the area in that map representing the fingers of piano players, those people play piano for a long time, like 30, 40, 50 years, they use their fingers. Their finger areas are much bigger in the homunculus than normal person. Use-dependent expansion, plasticity. Okay? If you don't use, that area will shrink. Okay? If I cut, and that was a kind of cruel experiment I had to do. So the raccoons, they use these two fingers a lot. So if you open the brain of a raccoon, you will see these two fingers, particularly these two fingers are heavily represented. You can easily see the structures. Now, what I did, I denervated those two fingers. So no signal will go to the brain from these two fingers. And then after a month or two, when we studied that area shrank, got shrink a lot because no signal is going there. Just opposite of piano player. That person uses the finger a lot, that's why that area got bigger. If you don't use, it will get shrink. Okay, so that use plasticity is present in these maps. And you can do many studies on it. It's interesting. Okay, so those are homunculi. If you visit the British Museum of History, you will see two guys, motor and sensory homunculi are kept there. Okay. Okay, so anything else we have here? No, that's all. Do you have any question? Anything? Any interesting question?